Today I want to talk about what rats can teach us about human brain health. My name is Keelan. I'm a PhD candidate at the University of Rochester, and I study fluid flows in animal brains. When you sleep, a fluid called cerebrospinal fluid, or CSF, circulates around and through your brain tissue, following this diagram that you can see here. Eventually, the CSF exits your brain through your neck and your skull, and scientists call this circulation process the glymphatic system, and think that it might be really important for maintaining brain health. One of the interesting pathways that CSF can move through is called a perivascular space, which is a tiny channel that surrounds blood vessels in the brain. On the left, you can see a 3D reconstruction of a real artery and a real perivascular space from a rat. We think that the flows in these channels in particular help get CSF deeper into brain tissue, so it can sweep away waste products, including the proteins that build up when patients have Alzheimer's disease. But most of what we know about these flows comes from experiments that were done on mice, and mice are not the same as humans. Our team wanted to see what would happen if we looked at rats, which are more similar to humans because their brains are more complex, they are bigger, and they are smarter. But crucially, they're still similar enough to mice that we can use the exact same experimental techniques and have a really good comparison of fluid motion in the two species. So how do we study fluid flows in animal brains? We use a technique called particle tracking, where we have a dye that lets us see the artery, and then a few bright particles that we can keep track of from frame to frame of a video, and stitch them together, which is what these sort of bright lines are, to figure out how fast the fluid is moving. This is the same thing that you're doing when you watch twigs or leaves float down the river, and you're able to figure out how fast the river is flowing. Previous experiments had seen that in mice there is this sort of steady flow in the same direction as blood, but there are also these pulsations with the heart rate. And we see something really similar in rats. Although the pulsations look a little bit different, they're still locked to the heart rate, and the fluid is still generally in the same direction as blood flow. When we directly compare the flow speeds, looking at several different locations in a few different rats and a few different mice, we found that the speeds are nearly identical in both species. But the flow speed only tells us part of the story. The other important thing to look at is called the flow rate. The flow rate is calculated by multiplying the average flow speed by the area of the channel that the fluid is moving through. You can think of this as highways with more or less lanes. Even if every car is traveling at the same speed, if you have 3 times as many lanes, you're moving 3 times as many cars. In animal brains, the perivascular spaces are kind of like the highways, and the speeds that we just measured are telling us a speed limit. But since the thing that we really care about is the rate at which a waste product would move, which is like the rate at which the cars move, the volume flow rate is going to tell us how much waste we can get out of the brain. And we found something interesting here, which is that rat perivascular spaces are actually much larger than the perivascular spaces in mice. So even though the flow speeds are similar, the rats are actually moving about 3 times as much fluid per second through these channels. Intriguingly, rats also produce about 3 times as much CSF each day compared to mice. And so the way we interpret that is that although not all of the CSF that your brain produces ends up in perivascular spaces, we think that the same set of possible fluid pathways exist in both rats and mice, and probably the same proportion of CSF is being directed through each of them. So we started by asking the question of if flows are the same in mouse and rat brains. And to figure this out, we performed the first ever high-resolution measurements of CSF flows in rats. What we found is that the flow speeds are similar, but the volume flow rates are actually much larger in rats. And there are several other differences as well. We looked a lot at things like artery pulsation and how that's affected by anesthesia in the 2 species. So if that sounds interesting to you, if you want to learn more about these pulsations and what they mean, you can check out the full paper or the lab website at the QR codes below. Lastly, I want to thank the funding agencies that gave us money for this project, and thank you for listening.